Transposable elements in bacteria. Bacterial transposons move within and between chromosome and plasmids are the extracircular chromosomal structures which are present in the bacterial cell. Bacterial TE contain and spread of antibiotic resistance gene. Bacterial transposons, insertion sequence or IS element will be there. Composite transposons, non-composite transposons. Bacterial transposons are demarcated by inverted terminal repeats. Then they insert into a DNA molecule. They create a duplication of sequence at their insertion site, a target site duplication. IS elements, insertion sequence or IS elements are the simplest bacterial transposon, smallest DNA fragment found in bacterial chromosome and plasmids. IS elements were first detected in certain lac negative gene mutations of E. coli. It reversed the wild type phenotype. IS elements are compactly organized and contain only genes whose products are involved in transposition. IS elements encode transposase, an enzyme. Insertion sequence or IS element are cut and paste transposons excision that reside in bacterial chromosomes and plasmids. IS elements can mediate recombination between different DNA molecules. Then conjugative plasmids can move transposons that contain genes to antibiotic resistance from one bacterial cell to another. Insertion sequence elements. Gene encode gene transposase for mobilization and insertion. Range in size from 768 BP to 5 kb. IS1 first identified in E. coli's galactosoporone as 768 base pair long and is present with 4 to 19 copies in the E. coli chromosomes. Ends of all known IS elements show inverted terminal repeats or ITRs. Insertion sequence IS1 transposase gene. Structure of IS50 element. Terminal inverter repeats. Target site duplication. Transposition of insertion sequence elements. Original copy remains in place. New copy inserts randomly. Transposition requires transposase copied by the IS element. IS element otherwise uses host enzymes for replication. Transposition initiates when transposases recognize ITRs or inverted terminal repeats. Site of integration is equal to target site. Staggered cuts are made in DNA at target site by transposase. IS element inserts. DNA polymerase and leakase fill the gaps. Transposase behave like a restriction enzyme. Small direct repeats, 5 base pair flanking the target site are created. Then structure and integration of IS element in chromosome DNA. Insertion of IS element into chromosomal DNA. Target site chromosomal DNA. Inserted IS element. Gaps filled by DNA polymerase, DNA leakase. Duplicated target site sequence. Insertion of an IS element causes target site duplication. The two strands of the target DNA are cleaved at different sites or arrows. This IS element is inserted into the gap created by staggered cleavage of the target DNA. DNA synthesis, dark blue color, fills in the gap on each side of the IS element, producing a direct duplication of the target site. Two different ways to cut DNA by restriction enzymes: blunt ends and overhanging ends, sticky ends. Then multiple IS elements. The bacterial chromosomes may contain several copies of an IS element. Plasmids may also contain IS element. When a particular IS element is found in both plasmid and a chromosome, homologous recombination may occur. Conjugative R or resistance plasmids. A non-chromosomal circular DNA exchanged during bacteria mating, which provide drug-resistant conjugative R plasmids, have spread multiple drug resistance in bacterial populations. These plasmids have two components. The resistance transfer factor or RTF contains genes required for conjugative transfer between cells. The R determinant contains the genes for antibiotic resistance. Formation of conjugation R plasmid by recombination of IS elements. Non-conjugative R plasmid, conjugative plasmid, R determinant, RTF component, conjugative R plasmid. Then complex bacterial transposons. Composite mobile genetic elements that are larger than IS elements and contain one or more protein coding genes in addition to those required for transposition. Non-composite mobile genetic elements are those which lack IS elements on its ends. Then composite transposons. Composite transposons are bacterial cut and paste transposons denoted by the symbol Tn are created when two IS elements insert near each other. Composite transposons consist of two IS elements flanking a region that contains one or more genes or antibiotic resistance. Composite transposon. A composite transposon is flanked by two separate IS elements which may or may not be exact replicas. Instead of each IS element moving separately, the entire length of DNA spanning from one IS element to the other is transposed as one complete unit. Genetic organization of composite transposons. Chloramphenicol, kamycin, kanamycin, streptomycin, tetracycline, bleomycin. The above antibiotics are protein synthesis inhibitors affecting the binding of amino-acyl tRNA to the mRNA ribosome complex. Non-composite transposon. Non-composite transposons which lack flanking insertion sequence. In each case, transposition requires specific DNA sequences located at the ends or a multi-unit complex. Encode transposition proteins have inverted repeats at their ends. In addition to resistance and virulence genes, they may encode catabolic enzymes. TN3 elements. TN3 elements are larger than the one IS elements. TN3 elements, like composite transposons, contain genes that are not required for transposition. TN3 elements have simple inverted repeats at each end, not 50 IS elements. TN3 elements produce target site duplication when they transpose. Genetic organization in gene TN3. TNPA transposase, resuscite, resolvase repressor, beta-lactamase, 38NP inverted terminal repeats. TN3 is a replicative transposon that transposes by temporarily using DNA molecules into a cointegrate. When the cointegrate is resolved, each of the constituent DNA molecules emerge with a copy of TN3. TN3 has 38 base inverted terminal repeat and contains 3 genes in its central region. One of those genes, BLA, encodes beta-lactamase, which breaks down ampicillin and therefore makes cells containing TN3 resistant to ampicillin. The other two genes, TNPA and TNPB, encode the enzyme transposase and resolvase that are needed for transposition of TN3. Transposase catalyzes insertion of the TN into new sites, and resolvase is an enzyme involved in the particular recombination events associated with transposition. Resolvase is not found in all transposons. The genes of transposition are in the central region for non-composite transposons, while they are in the terminal IS elements for composite transposons. Non-composite transposons also cause target site duplications when they move. So transposition of TN3. The transposases encoded by TN3 catalyzes the formation of a co-integrate between the donor and the recipient plasmids. During this process, TN3 is replicated so that there is a copy of the element at each junction in this co-integrate. Resolvase produced by the TNPR gene resolves the co-integrate by mediating recombination between the two TN3 elements. Donor and recipient plasmids separate, each with a copy of TN3. Association, replication, integration, and resolution and transposase integrase will be there. Transposable elements in eukaryotes. Transposons in maize, P elements in Drosophila, Sine ALU family, line L1 of humans. Transposable elements in eukaryotes. Barbara McClintock in McClintock in 1902 to 1992, Cold Spring Harbor Laboratory, NY. Nobel Prize in Physiology and Medicine 1983 for her discovery of mobile genetic elements. Studied transposable elements in cone 1950s, formerly identified as mutated genes by Marcus Roades, also known for work demonstrating crossing over as part of chromosomal basis of inheritance. General properties of plant transposons: possess ITR sequences and generate short repeats at target sites. Many activate or repress target genes, cause chromosome mutations, and disrupt genes. Two types: autonomous elements transpose themselves, possess transposition genes. Non-autonomous elements do not transpose themselves, back transposition gene, and rely on presence of another TL. McClintock, MC Clintock. I will be pronouncing it as like that. I don't know what is pronunciation. MC McClintock demonstrated purple spots in otherwise white Gonzi maize kernels are results of both these types of transposable elements. Like this. McClintock's discovery of transposons in corn, white kernels, and C- purple kernels. Kernel color allele traits are unstable. If reversion of a C to capital C occurs in a cell, cell will produce purple pigment and a spot. Earlier in development, the reversion occurs, the larger the spot. McClintock concluded C allele results from a non-autonomous transposon called DS inserted into the C gene. Disassociation. Then autonomous transposition AC controls DS transposon. AC activated. ACDC system transposon effects on cone kernel color. Purple kernel, normal C gene expressing pigment product. Colorless kernel activates DS transposition. DS can transpose into C. Disrupted or mutant C gene. Then spotted kernels activates DS transposition out of C in a few cells during kernel development. Reversion of C mutation of capital C. Mutation G. CG. Normal CG. MC Clintock's discovery of transposons in corn. AC elements is autonomous, DS element is non-autonomous. AC activates DS. DS varies with length and sequence but possesses some ITRs and AC. Many DC elements are detected or rearranged versions of AC. DS element derived from AC. AC-DS are developmentally regulated. AC or DS transpose only during chromosome replication and do not leave copies behind. Structure of AC autonomous and DS non-autonomous transposable elements in corn. Activated element, intron, exon, and transcription will be happening. Then dissociation elements are shown. Transposable genetic elements. Transposable elements and overview, definition, features and types. Transposition and mechanism in bacteria, IS elements, TN family, MU phage as a transposable element. Then in eukaryotes, transposons in maize, P elements in Drosophila. So transposable elements in human, sign ALU family line and L1, retrovirus and retrotransposons. Transposable elements, mobile genetic elements, contribute to spontaneous mutation, genetic rearrangement, horizontal transfer of genetic material, aid speciation and genetic change in bacterial. Transposons are often associated with antibiotic resistance genes. Cells must depress transposition to ensure genetic stability. Transposable elements are transposons. 40% of the genomic DNA. They are specific sequences of DNA. They are found in the genomes of many kinds of organisms. They are structurally and functionally diverse. Transposition, insertion, mutation. Definition and keywords. Transposons are sequences of DNA that can move around to different positions within the genome of a single cell, a process called transposition. Transposase, an enzyme that binds to ends of transposon and analyzes the movement of the transposon to another part of the genome by a cut and paste mechanism or a replicative transposition mechanism. IS elements, a short DNA sequence that acts as a simple transposable element. Then transposase, inverted terminal repeat. In the process, they may cause mutations, increase or decrease the amount of DNA in the genome, promote genome rearrangements, regulate gene expression, induce chromosome breakage and rearrangement. Next, types of transposable elements, DNA versus RNA, viral versus non-viral, replicative mechanism versus excision mechanism. DNA versus RNA. DNA transposons. Transposons consisting only of DNA that moves directly from place to place. Retrotransposons first transcribe the DNA into RNA and then use reverse transcriptase to make a DNA copy location. First transcribe the DNA into RNA and then use reverse transcriptase to make a DNA copy of the RNA to insert it in a new location. In retrotransposition, RNA molecules that are reverse transcribed by an enzyme reverse transcriptase into DNA molecule. These DNA molecules are subsequently inserted into new genomic positions. Mechanisms of transposition. Two distinct mechanisms of transposition, like replicative transposition, direct interaction between the donor transposon and the target site resulting in copying of donor element. Then conservative transposition or excision, involving excision of the element and reintegration of a new site. Two methods of transposition, cut and paste mechanism, transposons, target site, interrupted DNA sequence. Copy and paste mechanism, transposon, target site, interrupted gene sequence. Mechanism of transposition, replicative transposition, copy of transposon sequence, transposon enzyme cut target DNA, transposition duplication of target sequence, TN3, bacteriophage MU. Then replicative transformation. In replicative transposition or copy paste, an element is replicated and one copy is inserted at one site. One copy also remains at the original site. Original cut of transposon is only nick and only one strand is initially ligated. Element indicates through itself, replicates through itself, produces as intermediate a co-integrate structure. Co-integrate is resolved by revolvase and at specific site. Non-replicative or conservative transposition. Cannot copy transposon sequence. Transition by cut and paste model. Cut transposon sequence from donor molecule attached to target site. Then excision transposition. In excision mechanism transposition, cut and paste, an element is cut off one site in a chromosome and pasted into the new site by an enzyme, the transposase, which is usually encoded by the transposon itself. T elements, cut and paste mechanism, cut themselves out of original site, producing double-stranded break, cut target site and ligate to element ends, thereby inserting at new site. Original site break repaired, usually with sister chromosome, restoring transposon at original site sometimes, and healed without transposon can also be associated with deletion at excision site. So categorization of transposable elements by transposition mechanism, cut and paste transposons, replicated transposons, retrotransposons and retroposons. Replicative transposition. Single stranded cuts are made on either side of the transposon and on the opposite side of the target side of. Single stranded cuts are made on either side of the transposon and on the opposite sides of the target of the recipient. Single stranded cuts produce staggering ends in both the transposon and target. This produces 4 free ends in each DNA molecule. 2 of the ends from the donor are ligated to 2 of the ends of target. This links the 2 molecules with a single molecule of transposon. Crossover structure, strand transfer complex, nicked ends of transposon are joined to nicked ends of target. Replication from free 3' end generate co-integrate. Single molecule has 2 types of transposon. The 2 remaining free 3' ends are used as primers for DNA polymerase, which uses the transposon DNA as a template. This replicates the transposon and leaves the conjugates.